Yeah, it's a big pleasure to be in this really unique meeting. I have never come to such a meeting before. So I'll try to narrate a little bit of the things that happened in the course of my journey for research, which will be more typical for the Indian situation, because when I started, when I was at the position that I had to set up my own lab, that was in 1982. I had done my postdoc in the US, and then I came back, and my training was in gene regulation. So then, and I had worked basically on bacterial systems. So I was looking for something which was more exciting and a new system to work on. And initially, since my BSc was in botany, so I was thinking of something like Wallbox, which was a very fascinating system. But the job that I got was in a medical institute. So then I had to look at some medical kind of relevant organism. And at that time, some papers were coming out on the kinetoplastids like Leishmania and Trypanosoma, which had some really fascinating molecular biology in those organisms. So for example, there was the transplicing of leader sequences to generate translatable mRNAs, like in the Leishmania, you have very large polycystonic transcripts, and these are not translated, but they are processed by using spliced leader RNAs. So those were very fascinating things. And then the extensive RNA editing by uridine addition and deletion, which was also discovered at that time. So like this is so fascinating that you make a transcript, but it's not translatable until you insert all these U's which are in red. So I thought that these organisms would be something really fascinating, and if I'm going to take the plunge to do some molecular biology, I might as well work on some exciting system. So this was in the early 1980s, and the refrain was from our seniors that it's not possible to do good molecular biology in India, because we had to literally import every reagent, and the paperwork involved, because we had to pay customs duty on everything. So not only was it doubly expensive, but also the paperwork was just humongous. Fortunately for me, I had done my PhD in India, so I sort of had an idea of the problems that I'm going to face. So I was quite realistic about it. So at that time, when I was trying to figure out what to work on, I met Shiv Pillai, and I mean some of you might know him. So he had just done his MBBS from CMC Vellore, and he had joined Kothari Center for Gastroenterology in Kolkata, and he was looking at this protozoan parasite Entamoeba histolytica, which was very prevalent especially in the eastern part of the country, and it causes amoebiasis. It had a relatively simple life cycle. So you just ingest the cyst which is there contaminating our food and water. Even today we have a lot of Entamoeba cysts. If you don't drink bottled water and you just take tap water, you will get Entamoeba cyst definitely. So once you ingest it, then in the intestine it gets converted to the trophozoite, and this is a very highly phagocytic cell, and it literally causes ulcers by contact dependent lysis of the cell that it comes in contact with. Then it can also go to the liver and form liver abscess and so on. So Shiv was working with these patients, and he had patient Sira, and he said, it's a very fascinating system, nobody is working on it in India, and even abroad the amount of effort was very less. So it would be a good thing to work on. But then if you want to do some molecular biology, and this is before the PCR was there, we needed some minimum amount of DNA to start working, and that means that you have to grow the parasite in a pure state. And then we realized that in India actually nobody was growing Entamoeba, nobody knew how to grow the parasite to the amount and in a pure enough state that you can use it for any molecular biology. Because in the intestine it is growing along with bacteria and other protozoa, and most of the work was being done in in the hospitals, and so they were just culturing a little bit in a mixed culture for microscopic examination or serodiagnosis. So that was not good enough. Now there was only one lab in the world, and that was at NIH in USA with Dr. Louis Diamond. He was a person who very laboriously had set up medium, this TYS33 medium, in which Entamoeba grows in an axenic manner. And Dr. Diamond used to come to India very often to collect samples because we had all the patients. So, he would take the fecal material from India and go back to NIH and then culture it, and he had a huge collection of Entamoeba strains from all over the world. So, then we met Dr. Diamond, and then we managed to get some funding, because to go to his lab he did not have any funds because all he was doing was culturing. He had not done any molecular analysis with these cultures. So, in any case, so then it was possible to go to his lab back as a postdoc and learn how to culture the parasite. So, at that time when I went to Dr. Diamond's lab, he said, okay, I can give you the amoeba cultures, but all I want to tell you is that it's really difficult to get DNA out of these cells. So, I said, how do you say that? Because he said, there's Diane Wirth. There was a very hotshot molecular parasitologist in Harvard Medical School. She was working on leishmania, but she got interested in Entamoeba also. So, she would take the morning flight from Boston and come to NIH, and Dr. Diamond would have the cells ready for her, and then she would get the DNA and go back. And it just wasn't working out. So, Dr. Diamond told me that, see, Diane has been trying to do it and she's not getting good preps. So, it was good that he warned me, because then I realized that this bug has a very potent nuclease. So, in fact, the moment you lyse the cells, you can almost see within seconds the viscosity of DNA coming down, because nucleases get activated. So, because I was forewarned, so it saved me time, because this was just a 6-month stint, and within 6 months I had to do a lot of things. So, it saved me time. And so then he had, of course, the whole collection of Entamoeba isolates from all over the world and other Entamoeba species also. So, I purified some DNA and did just a blot using different restriction enzymes. And what I found is that there is some highly repetitive DNA in this, because I am getting all these bands which are coming just with total genomic DNA. So, we know that one of the repetitive DNAs could be a ribosomal coding for ribosomal RNA. And at that time, of course, we did not have any sequence available or anything like that. So, I had to use heterologous probes. So, I took some probes from a malaria parasite which was available in that lab. And so, we showed that most of these bands were actually coming from ribosomal RNA, except this one band C, which did not light up with the ribosomal RNA probes. So, I will talk about that band C a little bit later. So, now it is okay, by repetitive DNA being ribosomal DNA is nothing very it is expected, but what we found in that short stay at NIH, I realized by using a battery of restriction enzymes that there was circular permutation to these molecules. So, then it gave us an indication that these are actually circular molecules, and that was interesting, because most of the time the ribosomal RNA genes are very safely there on the chromosomes, linear chromosomes. They are not, because plasmids can be lost by the cell. So, you do not generally find them an extra chromosomal situation. So, that was exciting. So, now I set up my lab in JNU. At that time I got a job in JNU, this was 1986. And I set up my lab, and then I was determined that I am going to work on this parasite, because it has already given me some fascinating clue about these extra chromosomal circles. So, of course, when you have an extra chromosomal circular molecule, you ask the question that does it have a fixed origin of replication, something like the bacterial plasmids. So, now this is where because I was back in JNU with very meager facilities, and I had to use a technique that I could work with very little resources. And at that time I came across this technique by Brewer and Fangman, which used 2D gels, and this was basically just agarose gel electrophoresis and southern blot. So, it was very doable within the constraints that I had in JNU. So, but these are very tough gels, so it is not easy to make them work. So, then I was lucky enough to have a student who was really brilliant, and not just the mental brilliance, but he had the perseverance and the tenacity to run these gels. So, like it looks simple enough that you run a 2D gel and then you do a southern hybridization, and then you can see that these are the arcs that you get when you have a replication bubble. So, this arrow shows a replication bubble. But the problem that we faced were because we needed radioisotope, that means we needed the nucleoside triphosphates radioactive, and no one in India was making it. Now, of course, we have Jonaki and CCMB from where you can purchase it. So, we had to import the radioactive nucleotides, which was really quite a hassle, because they have a short half life and then you needed lot of clearances. So, those are problems that are typical to India, and I am glad that many of them have now been sort of solved, but although we still are plagued by many of those, and we still have to work hard to solve them. Actually, there is a story also that how when we had to pay customs duty to import chemicals, actually one of the scientists happened to meet the then Prime Minister Rajiv Gandhi, and Rajiv Gandhi took personal interest to say waive off that. So, he gave us a custom duty exemption certificate, which we all used to be very happy to get. So, by the 2D gels, then it kind of opened up the picture for us that these molecules actually do not have a fixed replication origin. So, this is something like the situation which we find even in eukaryotic chromosomes, where you have many potential origin of replication, but they are you do not have fixed origin, you have many potential origins, which are then some of them may be activated at some times. So, what was surprising is that in Entamoeba, actually even a small plasmid, which was only 25 kilobase, also had these multiple origins of replication. And then later on we could also show it by electron microscopy, and we could map the ories by digesting this molecule with BSA H1, which had a single site. And then so, after that student who really managed to get it, I had one more student who was strong enough to take it on, and Shoma, she then showed that these replication origins we could kind of synchronize the cells by serum starvation. And so, the replication stopped when you serum starved the cells, and then when they reappeared, at that time you could see that the multiple ories were not used all at the same time, but there were some early origins of replication and there were some late ones. Okay. So, now it was kind of getting difficult for me to understand the replication story deeper, because at that time we did not have the sequencing project of Entamoeba was not yet there, and so it was really difficult to like biochemically to purify enzymes which are involved in replication. That was not possible because we could not grow the cells to those large numbers, and PCR etc., it was before those days. So, then I had to kind of, as I said, that most of the time we were doing things which were possible to achieve at that point. So, it was then possible to think about transcription. And so, we asked the question that where are these, where is the nucleolus in Entamoeba? So, we found that we could get antibodies against fibrillarian of Entamoeba and also against one of the RNA polymerase 1 subunits. And so, already people had shown that just if you stain by iron hematoxylin or iodine, you can see that there are two fixed regions which stain very prominently in the Entamoeba nucleus. One is the peripheral chromatin and the other is the endosome. And when we use these antibodies, then you can see that it is a peripheral chromatin which is lighting up with the fibrillarian and also with the polymerase 1. And so, it is a peripheral chromatin which actually is a nucleolus in Entamoeba. So, that is something that we could determine. Then we wanted to look at the regulation of ribosomal RNA during growth stress. Well, that was something which easy enough question, and it could give us some good information. So, it was known that in the model systems like yeast and mammals, whenever there is a growth stress, then ribosomal RNA genes the transcription is stopped, and people were trying to work out how this transcriptional control is happening. So, we started looking at it, and in Entamoeba actually what we found is that this control is not at the level of transcription, but it is after transcription. So, do not want to go into all the data, but so basically the growth stress that we did was serum starvation, because these cells need serum for their growth. And what we found is that when we serum starve the cells, actually pre-ribosomal RNA is accumulating in these cells, and also some of the intermediates accumulate in the cell. And then this is of course more recent data. So, so then we know that there is an enzyme encoded by the gene RRP6, which is involved in the processing of pre-ribosomal RNA. And this enzyme, it is an exonuclease. So, in normal cells it is there in the nucleus, but when we serum starve the cells, then it is lost from the nuclei. So, that may be the reason why, although the transcription is going on after serum starvation, because the exonuclease exits the nucleus. So, therefore, these transcripts accumulate. And we found the same thing happens even with ribosomal proteins. They are transcribed, but they are not translated. So, like here these white bars are the transcripts. So, during serum starvation the transcripts are there, but they are not translated. So, it is a post transcriptional effect. And we also could show that this translational down regulation is mediated through the UTR. The UTRs in Entamoeba are very short, only 10 to 100 nucleotides. And so, these nucleotides, when we mutated these just upstream of the AUG, what we found is that in this mutant the translational down regulation is actually then relieved. So, so that means that in Entamoeba, whenever it undergoes stress, it does not really want to shut down its transcription. It wants to maintain the pools of ribosomal RNA and ribosomal proteins, and the logic of that is because Entamoeba actually has lost the ability to synthesize purines. It has to take up purines from the outside milieu. So, therefore, if it has to now synthesize ribosomal RNA and proteins when the stress conditions are relieved, then it will probably be incapable to match the need for new ribosomes, which will be very rapid. So, therefore, it helps the parasite to have stores of ribosomal RNA and ribosomal protein mRNAs, and then when the stress is relieved, it can process the pre-ribosomal RNA which is lying in an unprocessed state, and for ribosomal protein mRNAs, then it can go into translation. So, that was the logic that we could derive from it. Then quickly, so there was this other band that at that time we did not know what it is. So, at that time actually one thing which we could do was Sanger sequencing, because that was again just running gels, and of course, we needed the isotope import and all, but we had been able to succeed in that. So, we had sequenced it, and then we were waiting because there was no BLAST algorithm at that time, and the databases were not there. So, after I do not know, maybe 7 or 8 years of our sequencing it, finally when the databases were there and we learned how to use BLAST. So, then we knew that this actually codes for a reverse transcriptase, and that reverse transcriptase is of the type which is there in the non-LTR retrotransposons, like the human EH lines and EH signs, ALU elements. So, it belongs to that family, not the LTR types like in retroviruses. So, that meant that Entamoeba actually has non-LTR kind of retrotransposon in its genome, and nobody had suspected that. So, it was quite exciting at that time. And then at that soon after we had made, not soon after, but after about 5 years when we did all the Sanger sequencing of these retrotransposons, finally there was some international funding, and Sanger Center and NIH collaborated to start the Entamoeba genome project. So, in 2005 then we finally got the Entamoeba genome sequence, and after that we could then kind of validate the data that we had very laboriously collected by Sanger sequencing, that these lines and signs, their organization in the Entamoeba genome. So, so then I wanted to spend some time to study the biology of these elements, that how does this movement take place. So, this is just to show that the lines and signs are very abundant in this genome, and there are 3 families. So, the family that EH line 1 and sign 1 is the most common, and the line 1 family it codes for there are 2 ORFs, So this is quite similar to even how it is in humans. The human line 1 also looks like this, with of course some differences. So there is a reverse transcriptase which is coded, and there is an endonuclease. This endonuclease needed to cut the target site when the element moves, and ORF1 is like a nucleic acid binding protein. It forms an RNP particle, and the SINE does not have any coding capacity except it shares some sequence at the very 3' end. All this we knew from the sequence analysis. So now the problem is that most of the line copies we found are actually inactive because they have accumulated mutations, and this is a way by which these elements survive in the genome, because if they are all the time active, then they will be highly mutagenic. So even in the human system we have millions of copies of these elements, but they are inactive. So we also found that from the database we could not get actually active copies. So that means if I want to study this process in Entamoeba, I have to make a cell line which will be able to express the functions needed for transposition. So that was quite a gamble, because it is a tough thing. Entamoeba system transfection and all was not easy, and the success rate was low. So now fortunately what happened, this endonuclease was sort of very easy to express in E. coli and to study its properties, the target site, where does it cut and all that, and do some kinetics. So I told the student that okay, let's do this, it will give you short paper, and after that we do the gamble. We try to express this in the whole ORF2 in Entamoeba and see whether we can notice retrotransposition. So he agreed with that, and so then we made cell line in which, so what we found is that ORF1 is actually constitutively expressed in Entamoeba. So we do not have to express that, only ORF2 we need to express. So we made cell line which was expressing ORF2, and then we tracked the movement of a line element in this cell line. So like now this plasmid contains a line, contains a SINE element in which we put a tag, so we can differentiate it from all the hundreds of copies of SINE which are there in the genome, and in this cell we are expressing ORF2. So, so then this SINE will be now transcribed to give a transcript, and if it is retrotransposition competent, then it might form an RNP, and this will go into the target site. So we had also provided the target site, which we had learned from other experiment that it is a possible target site, and now it will insert into the new target site. So we will have sort of another copy of this tagged SINE in this plasmid, and this is something which we can measure by PCR. So I am not giving the data, but then we showed that by PCR that it actually moves to the new site. Then the question that we asked is that okay, when it moves to the new site, I mean I am just saying all this because it didn't need high technology. So just trying to show you that if you just go on with whatever technology is available, you can still do some interesting, you can get some interesting answers. So the question that we then asked is that when this SINE element is transcribed and then it gets reverse transcribed into the new site, does it just go directly as such unchanged, or does it suffer any changes? And this question was important because reverse transcriptase is a much more error prone enzyme than DNA polymerase. So we know that reverse transcriptase does introduce changes in the molecules during the process of reverse transcription. So what we did again just by PCR, so this new copy which has moved to this site, we just PCR amplified from the flanking regions and cloned these and then sequenced 23 randomly picked clones. And what we found is that out of these 23, 10 of them actually had the original tag the way we had introduced it, but 8 of them were the copies which were there in the genome, because genome has hundreds of copies and they are also getting transcribed and they also get mobilized by the same system. But interestingly, there were 5 of them which were recombinant SINEs, and these 5 probably have arisen because the tag has moved into some of these genomic SINE copies. So they have the tag, but they do not have the original sequence that we had supplied. So it is a process of recombination which is not at the DNA level because it is highly rapid, and we showed that it only happens when the ORF2 has been provided in the cell. So, it is not happening just because the tagged sign is sitting in the cell and okay. So, basically then I mean we have a hypothesis for it that when the RNP particle forms, then actually the reverse transcriptase, it switches the template, and that is how you get the recombinant molecules. So, so that now I am almost coming to the end of my Entamoeba journey, because I am going to superannuate, and you know, Entamoeba is a very tough system to grow and maintain in the lab. It requires dedicated funding, and you know, we do not have in India postdocs that much, especially in universities we do not have much postdoc and technicians. So, it is driven by students. So, now that I will not have students, so I decided that Entamoeba I will not be able to carry it forward. But then I am going to put some effort in a new system, and this is GNE myopathy, a rare genetic disorder which is there in my family itself. So, it is like a vested interest. So, I am basically going to have a mentor role sort of a thing, and just do the thought process. So, I was talking in fact to Jyotsna also. So, we will try to carry it forward, and so far whatever little bit we have done, we already found that there is one mutation in this gene which is very, very India specific. It is only found in South Asia. So, even in the 1000 Genomes project, when we looked at the data, this particular mutation is found only in the Indian samples. So, it will be really interesting to even to look at you know, how these kind of mutations have arisen in the Indian diaspora. So, that is all. Okay, yeah, I must end by thanking all my students, because they are the ones who did all the work. As I said, we do not have postdocs and technicians in India. Everything is driven, I mean not in India, I think institutes are little better, but in university system it is all driven by your own funding. So, you have to depend on students, and these are all the students who have made possible for me to tell you whatever I have done today. Thank you.